Hello, this presentation will provide an overview of Aldevron's process development and manufacturing for RNA and protein-based therapies and vaccines. I'm Nate Spangler, Director of Innovation and Strategy for the RNA platform, with my colleagues Tom Lynch, Vice President of Client Services for our Protein Business Unit, and James Sulzberger, Technical Director of Client Services for the Protein Business Unit. They will cover our capabilities to supply enzymes for RNA manufacturing and other proteins for vaccine development. We thank you for taking the time to watch. Long forms of synthetic RNA, such as messenger RNA and self-amplifying RNA, have the potential to transform medicine. Manufactured in a cell-free process by in vitro transcription, this new generation of drug delivers transient genetic information, which enables the body to make its own medicine or vaccine in the form of the enzyme, antibody, or antigen that is encoded by the RNA. The possible range of applications is broad, and we're seeing promising potential in experimental therapies and rapid responses to emerging pandemic threats. Excited by the possibility that this new generation of drugs will fill critical gaps not met by traditional small molecule drugs, available biological treatments, and emerging gene therapies, Aldevron is building a comprehensive manufacturing platform for messenger and self-amplifying RNA that spans preclinical to commercial stages of production. This is backed by decades of experience manufacturing plasmid DNA and recombinant proteins for the biotech and biopharmaceutical industries. We have extensive experience manufacturing for many uses: drug products, drug substances, raw materials, and ancillary materials for gene and cell therapies, protein replacement therapies, cancer vaccines, infectious disease vaccines, and more. Founded in 1998, our main campus and headquarters are in Fargo, North Dakota, where we are completing construction of a 190,000 square foot addition to the new 70,000 square foot manufacturing facility that opened in September of 2018. Our 5-year plan is to develop up to 500,000 square feet to support preclinical to commercial stage manufacturing of DNA, RNA, and recombinant proteins. Our expansion plan was designed in consultation with regulatory agencies, industry professionals, and most importantly, our clients. Design decisions promote efficiency and scaling and the next generation of manufacturing optimization for patient scale to global scale production. Plans include a visitor center and 100,000 square feet of R&D and technical operations and training center. Currently, Aldevron has more than 450 employees across sites in Fargo and Madison, Wisconsin, where we specialize in recombinant enzymes for gene editing and RNA synthesis. With messenger and self-amplifying RNA manufacturing, every program has unique requirements for support and direction in reaching the clinic. And Aldevron has multidisciplinary teams of experts to understand client-specific requirements and to engineer manufacturing solutions that fit. An RNA program at Aldevron can begin with a DNA template design and the RNA use requirements. Aldevron can manufacture using our standard processes or client-specific processes that we develop, or a client process transferred to us. There are 4 stages to a new program, beginning with exploration or tech transfer, depending on the amount of process knowledge that is available and the readiness for scale-up. In the first stage, RNA manufacturing knowledge is gained to enable the scale-up work that happens in the second stage. A master cell bank is generated and the linear template manufactured, QC tested, QA reviewed, and released by the end of stage 2. A full-scale RNA engineering run is performed in the third stage, and resulting material is released under the GMP source quality level, which is an Aldevron feature I'll describe in a moment. At the fourth and final stage, QC test qualifications are completed, and manufacturing documentation is finalized and approved for GMP manufacturing, which concludes with the delivery of mRNA drug substance, manufacturing batch records, and a certificate of analysis. The GMP source quality level I mentioned on the last slide was created by Aldevron to bridge the gap between research grade and GMP manufacturing for an easier transition into clinical stages of development. A faster and more economical alternative to GMP production, GMP source can be manufactured to standard or customizable specifications. Adopting the most relevant features of GMP manufacturing, including traceability, document control, and material segregation, GMP source can be used as an ancillary or critical raw material for producing protein and viral candidates and messenger RNA used in clinical trials. Accompanied by a comprehensive manufacturing summary report suitable for regulatory submissions, we deliver high-quality products that fit your GMP source project. With research grade, GMP source, and GMP, Aldevron offers progressive quality systems designed to fit all stages from discovery to commercial. A typical RNA manufacturing program spanning the 4 stages described a moment ago can run 9 months or longer, but this timeline can be compressed for urgent needs. Because messenger and self-amplifying RNA can be synthesized using enzymes rather than cell culture, the manufacturing process is simpler, more scalable, and more easily expedited than for other biological medicines. This has been a key advantage for rapid responses to emerging threats and unmet medical needs. In response to COVID-19, for example, we're helping clients go from antigen selection to clinical testing in a fraction of the time needed for conventional vaccine technologies. However, we've seen that the ability to rapidly scale up vaccine manufacture can be limited by plasmid DNA template and enzyme supplies. We're also working to address these critical needs, which my colleagues Tom and Jim will talk about more in a moment. Aldevron's RNA production is flexible and offers manufacturing options to satisfy client preferences for capping and tailing methods and configurations. Our standard purification process involves ion exchange chromatography and tangential flow filtration, and the benefits are high downstream process recovery and scalability and high product quality. To satisfy the stringent purity requirements of some uses, we can incorporate known methods such as affinity and reverse phase chromatography. As mentioned, we offer custom process development, and we can scale up client manufacturing processes. And Aldevron manufactures RNA using our enzymes or commercially available sources that are supplied in animal origin-free or GMP grades. Currently, our clinical grade production scales range from 1/10 of a liter to tens of liters for the IVT reaction, and we're planning to support scales 10 times larger in our new facility coming online next year. This will include the ability to accommodate downstream processes with large volumes of flammable solvents, as well as requirements for BSL-2 containment. For RNA product testing and release, Aldevron offers a comprehensive selection of QC tests that range from compendial methods for critical contaminants to product-specific tests for identity, integrity, and purity that comply with regulatory requirements for product release. We have a range of capabilities, either internally or through qualified partners, to run the tests needed for quality assurance. A major advantage to partnering with Aldevron for your messenger RNA production is that we also manufacture the master cell bank and linear template in GMP source or GMP quality grades according to client preferences. Aldevron has more than 2 decades of experience manufacturing high-quality supercoiled plasmids satisfying the stringent requirements for use in linear template production. We have experience with a range of enzymes for linearization, and we understand the challenges of poly-A tail retention in plasmid template manufacturing. Aldevron can also provide, if desired, all enzymes needed for in vitro transcription, post-transcriptional capping, and post-transcriptional polyadenylation. Our stocked enzyme products are manufactured animal component-free and research grade, and we can make to order in GMP source and GMP quality grades. Research and GMP source manufacturing is supported by multiple fermenters with stainless steel trains up to 1,000 liter working volume and multiple single-use fermenters up to 300 liters that are dedicated to GMP source and GMP manufacturing. Enzyme development and manufacturing are the focus of my colleagues, Tom and Jim, who will take it from here.
For over 10 years, Aldevron's protein business unit has been helping scientists advance their programs in the field of vaccine discovery and development. And through those years, we've built up quite a bit of know-how and capabilities and capacity in the pursuit of being the premier CDMO for bacterially expressed proteins. And in support of our rapidly growing RNA business, the team here in Madison, Wisconsin, has been producing the enzymes needed for IVT reactions for our clients, partners, and most certainly our colleagues in Fargo, where the nucleic acids business unit is headquartered. Moving on to the next slide, showing a list of currently available enzymes that can be purchased as a complete set for the IVT reaction through capping, but also a la carte, as each enzyme can be produced in custom batches that match our clients' requirements in terms of quality and scale. Now, each of these enzymes are available as research-grade products with animal origin-free, TSE, BSE-free statements. And we are currently scaling our operation to include the manufacture of these enzymes at both our GMP source and GMP quality levels. Now, as Nate mentioned earlier, IVT enzymes are just another key component of the RNA manufacturing process and supply chain that Aldevron is able to control. This is something that we see as being critical as the demand for high-quality RNA continues to grow. However, we are not only making enzymes to support RNA-based vaccine manufacturing. We also have the capabilities to help advance programs where process development and subsequent custom manufacturing are needed for protein-based vaccines. From an electronic sequence to manufacturing process tech transfer, our scientists work to build robust and scalable production methods that can be utilized for research purpose through clinical applications. In Aldevron, our focus is on providing an excellent client experience. From the very start, our experts are available to meet and learn about your requirements and evaluate what is needed to advance your program from a resource, time, and technical perspective. Now, should work be needed on either the upstream or downstream processes, our experts will apply their knowledge and experience towards finding an optimal solution. And if your program has a set of robust procedures ready to go, we have the systems in place to allow for a seamless transfer into our manufacturing operation. Either way, it's through scientific execution that we're able to build trust with our clients and partners. Now, to provide further details on our technical capabilities and the ways in which we work with clients for the custom manufacture of their proteins and enzymes in support of vaccine development, I'd like to introduce you to Jim Sulzberger, Aldevron's technical director. Hello, Jim. I'll now hand the reins over to you.
Thanks, Tom. No matter how far you are in your pipeline, whether it's simply an idea in an electronic sequence or it's a fully developed manufacturing process, Aldevron's technical capabilities within can help you get to the finish line. One of our strengths at Aldevron is that we are inclined to be strictly focused on bacterial expression of recombinant proteins. Some such vaccines are subunit vaccine components, carriers, virus-like particles, and other recombinant proteins used for vaccines. Our upstream capabilities range from small-scale shake flask screening and clone selection to high-throughput screening, design of experiment, optimization, and engineering scale up to thousands of liters of fermentate to support your clinical needs. In addition to our upstream capabilities for generating the target protein, our downstream capabilities at Aldevron are paralleled by no other bacterial CDMO within the industry. Our process design is typically governed by quality by design, meaning quality of the target product profile is built into the design of the process rather than tested into it at the end. Using traditional modes of purification such as ion exchange, hydrophobic interaction chromatography, and affinity chromatography are commonly employed. However, as the need for more advanced targets to fight different illnesses arise, targets become more complex. We also are able to employ new modern modes of chromatography and purification, such as mixed mode chromatography, hydroxyapatite chromatography, and membrane-based chromatography formats where applicable to intensify processes. Additionally, as larger quantities are required, we use economic tools such as single pass TFF and inline concentration to ensure that the volumes remain as small as feasible within the manufacturing space. Both one factor at a time, and design of experiment methodologies using high throughput screening are employed to ensure an efficient, robust, economic process development and a well understood design space for manufacturing. At the cornerstones of Aldevron's protein manufacturing services, scalability and process economics are key and are something that are considered even upon the initial discussions with the client and understanding their goals. We have decades of industry experience in reviewing bacterially expressed protein processes and suggesting, if anywhere, where a process might be made to ensure that the process gets to the clinic as soon as possible. Thank you so much, Tom, for letting me speak about Aldevron's protein services, and I'll hand it back to you.
Well, thank you very much, Jim. I hope this was helpful and informative for our audience. We'll now send it back to Nate for some closing thoughts. Nate?
Since its first use in 1990 to express reporter proteins in mice, followed by the demonstration of antigen-specific immune responses, there's been strong interest in RNA technology as a versatile platform to span many therapeutic and prophylactic fields. In the past decade especially, technology advancements in RNA biology, chemistry, stability, and delivery systems have accelerated the development of fully synthetic vaccines and therapies based on messenger and self-amplifying RNA. This is leading to a new generation of treatments delivered by RNA encoding enzymes, antibodies, and antigens. With the first messenger RNA-based therapies and vaccines now in the clinic and proving their safety and effectiveness, we expect to see usage rapidly expand to include a wider and more comprehensive range of indications. Leading the way will be both therapeutic and prophylactic vaccines based on messenger and self-amplifying RNA technologies. Aldevron is preparing for the future by building comprehensive platforms and greater capabilities to support RNA-based therapies and vaccines from preclinical development to commercial manufacture. We have the world's largest GMP facility for plasmid DNA and messenger RNA, and plans are underway to increase capacity by more than tenfold over the next 5 years. As a manufacturer, Aldevron stands apart through its offering of all critical biological materials needed for messenger and self-amplifying RNA production. For 21 years, our mission has been to help improve lives through innovative partnerships and scientific excellence. Our process development and manufacturing capabilities support the innovative therapies and vaccines that will transform medicine. The biopharmaceutical industry is growing exponentially, and we are prepared for and excited about the future. I'd like to thank the audience for your time and attention, and my colleagues, Tom and Jim, for sharing their knowledge and insights on the critical supplies of materials for RNA manufacturing.